Hello, I'm Wesley Janssen. I've been working with Mike Stangl for 7 years now on the regenerative method of lawn or soil care. So I guess I'm going to be doing a live microscope demonstration for you today. The microscope we use is this OMAX one, compound microscope. We have a 4, 10, 20, and 40 times objective, all multiplied by a 10 times eyepiece. So we get a 40, 100, 200, and 400 view of each view we have. We also have this camera up top. It's a 2 megapixel, which is on the screen, which we'll be using later to show you the slides. So we use 2 different methods here, one by Dr. Ingham. First, we use basic slides. They're 24 by 76 millimeter, 1 millimeter thickness. Pick these up anywhere, along with cover slips, 20 by 20 millimeter. We can go bigger, but it's all changed in our calculations that we use for the biomass ratio. We also use Dr. Johnson's method, which uses these hemocytometer slides. They're usually used for blood counting, but it's been changed. We've used it for spore counting instead. This is a cheaper one that we bought before, just for playing around with, but it doesn't work very well because the grid pattern has all the lines crooked. So we've taken a soil sample from one of our Johnson Su Bioreactors, and we're going to take a look at it today. We'll start with just the basic Dr. Ingham's method. We mix our sample up, try to get a pretty well homogeneous thing, or homogeneous mixture. Usually we would measure it using just regular scale, but just to save time, I'm just going to wing it pretty well. So what we usually use is approximately 1 gram of soil. We use our water to dilute it up to 5 milliliters. Put the cap on. Give it a gentle shake 30 times. 6, 8, 10, 16, 17. Take 1 drop of this, put it on the slide, take our cover slip and gently put it on there. Now this isn't the best sample done, but it'll work. Now for regenerative lawn care, we focus on the fungus to bacteria biomass ratio. So usually we are looking for anything alive in the soil, the fungus, the bacteria, protozoa, nematodes, all that stuff. So for the first view, we're going to take, this is a cover slip, imagine it. Basically what we do at a 1:5 dilution is we go back and forth along the cover slip, trying to get the entire thing viewed. That we look for nematodes, arthropods, worms, anything of the larger nature that we can identify. That's what we do at the 1:5. Now usually at a 1:5 dilution it looks pretty bad. Like, we won't be able to see much once we go to lower magnifications. This is at our lowest 40x. So we usually just zoom right along, up and down, back and forth, all the way along the entire thing. Sometimes I'll mix it up and go left and right instead, just to have a little variety in life. And hopefully we'll get to see something today. If not, after I'm finished with showing off the slides, I'll throw up a few pictures or videos for you guys, just so you can actually see something interesting. Now what we're seeing is a lot of debris. These are all roots and just general organic matter. Nope, we actually got a nematode today. We usually zoom right in so we can identify what type it is. There are 4 different types that we focus on. They are all based on what they eat: bacterial feeding, fungal feeding, root feeding, and predatorial. The predatorial will eat insects and other nematodes. Now, I know the camera doesn't show it up very well, but I believe this guy is a bacterial nematode. So he is a good one we're looking for. Usually the only bad ones are the root feeding, because when they pierce the root, that could introduce disease and other problems to the overall plant. So that will get marked down in our spreadsheet that I'm showing off behind us. That's how we keep track of what we do, and it's a lot easier to do the calculations on there through Excel rather than by hand, because the numbers get pretty big. Well, let's just keep zooming along here. Seeing some roots, more organic matter, a few cysts, some seeds, and we'll get where the accidental water edge. This is supposed to be even with the cover slip, but I didn't do a very good job. Usually I try to do a good job and get the whole cover slip filled, but just for show today, we're just going to go with it. So, not much moving in this. It's just because of the weather. It's been pretty cold lately, so most of the most of the living things do go dormant during the winter. So it is a surprise we get to see that nematode wherever he got to. But what we focus on usually is, this is at our highest magnification, 400x. We usually try to count the bacteria in here, but as you can see, it's pretty messy, and you know, these little bacteria in here, we're not going to be able to count those very efficiently or correctly. So what we do in that case, grab our glasses, go back to our sample, and we dilute further. Usually we can get away with getting up to just 10 dilution. Just taking our dilution, bopping it up to 10 mils, giving it another gentle 30 shakes. And 30. Pop off, splash it a few times, and take a drop. Now this still looks a bit messy at this point, but hopefully it'll look clean enough so I can actually show you what we focus on. Come on, cover slip. Gently. That's a lot better. That's what it should look like, fully covered. Let's pop that back on there, and it's already looking a lot cleaner and a lot better. Still pretty thick, so it could be iffy if we can actually do a count on this. Now usually I would go over this again, following that pattern up and down all the way across, just to do my due diligence and make sure there's not like 20 nematodes all of a sudden in this where there's only 1 at 5 dilution. So we'll just go up and down again. Just do it quickly this time. Doesn't look like we're getting too lucky today. Usually we'd see more from our Johnson Su bioreactors just because of how much organic matter is in it and how active it usually is. But sometimes you just get bad samples where you don't see much. That's why when we usually do the testing, we do 3 rounds of the testing just to make sure we get a good view and make sure we don't just all of a sudden get a really bad sample and base everything off of that. No nematodes here. And then we just scooch ourselves down. This is our highest magnification again, a lot cleaner. We would be able to count this. It wouldn't be as accurate as I would like, so I would usually dilute a bit farther. But just for the sake, I'm sure I've gone over it twice. I'm sure you understand what I mean. Usually for farther dilutions, because of the size of the test tubes, I would take 1 milliliter of this, throw it in another one, and then dilute that up to 5 here. But we'd have to multiply that by 10, so it'd be 50 dilution at that point, just because 1 gram 10 divided 5, multiplied 5 times would result in 50 dilution. So that's getting pretty high, and we would lose accuracy with what we're doing, but in order to be able to do the count properly, we would have to dilute that high, please. Now we do have some fungus right here, nice strand, a little bit beat up. Not sure if you can actually get a clear view on that, but so usually we would just zoom around. We would do our 20 views, and if we can grab this paper here, imagine that as a cover slip again. What we would do, we would just start somewhere, not look for specifics, but we would start one view there. Then we would just randomly jump this way. Second view. Then we do another random jump over here, 3 views. And maybe if I feel like it, I'll go back up here, 4 views, and really it's just, we try not to go in a straight line or focus on a certain area. We're trying to get an overall view of the entire thing. And we'll do that 20 times, do the count on each section, add that all up, and get our biomass ratio. So, if we can see clearly here, all these little dots that are moving, those are all little bacteria. We've also got a spore right here. That's what we like to see, because that is potential fungus. Let's see if we can find anything interesting. We got an amoeba shell right there. It's not at home, so either it's somewhere else in all this sample. But keep zooming around. There's a few more spores, some fungus that I probably can't get very clear for you. I'm trying to find something, hopefully. There's some more fungus. Pretty nice to see. That is what we focus on. Usually we would count that. Maybe like count it as between 1 and 10 based on how long it is based on the view. So, and across the width would be based on, let's take 1 bacteria and measure that across. So there's an amoeba that's in its shell, clearly. A few more right there, actually 3 of them, or 2 that you can see. There's a third one down there. So, that's basically what we do. There's some more spores right here. And we take those spores and we can identify them by this giant poster that we've had printed up. Aim it up there. Usually that helps us identify what it is, whether it's harmful or good or just neutral in general. So we can see how diverse the soil is, really. And we are seeing quite a few amoeba in here, quite a few spores, some cysts. That guy right there, that's just a dormant, or one of the protozoal, amoeba, flagellate, or ciliate, gone dormant. They create this little protective shell around themselves just to survive harsh conditions, which otherwise they couldn't. But there's not much interesting in this sample. So Good. We'll just leave that there. Maybe something will float by or swim in there, and let's move on. Glasses back. So that was the method we usually use based on Dr. Ingham's tutelage. So this time we'll switch to what Dr. Johnson is instructing us to do for the spore counting. We use this little hemocytometer. This is the better one we have that the grid lines are straight on. You'll see that in the view. The cover slips we use for that one are 22 by 26 millimeter, 0.5 millimeter thick. That's just to fit correctly on this. And we can just use this same sample again that we've already, or no we can't. Never mind, sorry about that. This guy we gotta use, these bigger test tubes. So, for this method, grab our sample, and for this we put 2 grams approximately in there. We usually measure it out correctly, but just to save time, I'm winging it again. So I'd say that's approximately 2 grams there. And for this guy, we dilute a lot farther. And we use these larger test tubes because we need to dilute up to 50 milliliters with 2 grams. So we just fill that up almost all the way. Lid back on before I spill that. And we just, for accuracy, bring it up to 50 mils with the dropper. Just like that. And we lid that. And we do the same thing, 30 gentle shakes. 5, 6, 7, 8, 9, 10, 11. Okay, we got that shaken up. Set that aside a little bit. So we can set up this hemocytometer. Now for this guy, we got, bring it a little closer. Our viewing area is in this central area, these 2 pillars. We got to wet those down a little bit first. So, a water along there. That way the cover slip can get a good seal. Because we then take our sample and shoot it in through those 2 little canals beside the main viewing area. That way, because the cover slip's already on the pillars, it's a set distance between the bottom and the cover slip. That way it keeps everything easily manageable and reproducible. Okay. Bye-bye to this slide. And we'll pop this one on there. Now usually, when using one of these, you would just focus on the grid pattern. But because it has these great big trenches along the sides, where'd the other one go? Great big trenches along the side, which is where we put the sample into. I like to go through those and make sure that any worms or nematodes or anything of a larger nature didn't get washed into there. And so we can actually make sure they're actually in there. Just need to focus that. And we'll just go right along there. Nothing in that one. Again, just a bunch more organic matter, roots, etc. Cell structures. center one. There's an air bubble in there accidentally. That's still fine. There's the little trench runs. So now I'll go over the entire thing again, just to check for nematodes in the measured depth. And we didn't get lucky this time, so we get to see nematode in this one. But here's the grid pattern I was talking about, if you can see it. It's this whiter area. Zoom in a little bit. But this is the main thing the hemocytometer is used for. Using this grid pattern, we can keep track and do proper calculations just because of the set squares we can use to easily count. And there's one on each side of the middle part, so we can do 2 tests on the same sample at once. So for these ones, we focus on the spores, like I said. Zoom in. There you go. And this is 1 square of the grid, which we would count. We would count the number of spores in that. Zoom back out so you can actually see the grid. So we would count the number of spores in this 1 square, which I only see 1, I believe, which is right here. And we would keep track of the total number we find in this, move square to square. There's another one down here and there, so that's 4. Couple more right here, 6. There's 4 in there. There. These are all pretty well about the same spores. We would think they're agaricus spores. So it's not showing much diversity yet, but it's still showing that there is possible growth. Got 2 spores there, the bottom here, there, agaricus, and I believe the cercosporas. If I misidentify anything, don't be offended. This is a learn as you go basis, and if you think I've made a mistake and want to correct me, please feel free to contact us and let us know. So I can learn, you can learn, everybody can learn, basically. So we do that for each. We do the spore counting for each square. There's 16 in total. So I'm just gonna blaze through it. We got a amoeba there and a bit of fungus. So we take that count, add it into our spreadsheet over here, a different spreadsheet that we use specifically for the hemocytometer. And then we go over to the next corner of the grid, do that again for these 16 squares, separate count. And then bottom corner and bottom left corner, we do that for each 16 squares in the corners. Then we take the average of that and do a bunch of calculation, and we get a total spore count. And that will show us how diverse the soil is for life and what potentially could grow there. And we learned of that method just this past year, and we've been using it a lot. It helps show the potential of each soil that we have. So that's pretty well it. And that's what we do each time. If there's any soils you want to send us, feel free to do so. We'll get them tested for you. So yeah, that's basically it. I hope you enjoyed this demonstration. I'll pull up some pictures right now, just because we didn't find anything that interesting there. Was that that guy. So these are just some basic photos of things I found throughout the years. So what we have here is one of the root feeding nematodes. It's got the great big spike here that can easily pierce the root and allow it to eat off of that. Next we have a grouping of spores coming off of a single fungal strand. Those, I believe, would be just smut, rust and smut spores. We got a nice fungus there, another spore, potentially a spore as well. So I believe that one is an alternaria spore. Got a nice nematode there. Not fully zoomed in enough to fully identify it, but just because of the length and the tail shape, I would say it's bacterial. That's another root feeding nematode. You can see the little spike they use to pierce the root. This is an arthropod we found. We don't see them very often, but they're always exciting to see just because they're different, and they basically are cleanups. They eat decomposing organic matter and only show up when there's high amounts of organic matter in order for it to have lots of food, really. Got some more fungus there, nice strand. We got a mite. It's just a mite, they can show up anywhere. Decomposers. There's another mite, he's not as clear to see. And there's the feeding end of a nematode. I got a very good picture of that. It's a bacterial feeder. See the dual stomach, and just nice picture. More fungus. It's mycorrhizal. I didn't focus fully onto this, but I have it labeled as mycorrhizal because it interacts with this root system here and actually does a symbiotic relationship with it. So those are the best pictures I have. I have many more pictures. If you want to see more, just let us know. I can send them to you. But they're not as good. I'm not always good at taking pictures. So basically, for our soil samples, if you want to send them to us, our soil samples, we use these little snack packs that we buy at the dollar store. This is a lot of soil. We don't need that much. As you saw, I use 1 gram and 2 gram. But what I would recommend is getting sandwich size bags instead, larger ones, sending us a little bit extra soil, just because, there can always be human error, and I could spill a sample and have to restart, basically. So we take the samples usually using a T-bar sampler. I think my sampler's out in the truck right now, but it's a bit expensive and kind of redundant. You don't need that much to take a sample that you need unless you're doing many samples in a day like I do. Sometimes I'll even use this little guy. I found this at Lee Valley. It's an apple core. It allows me to just jam in the soil, take it out, and it's spring loaded, so we can easily take the sample out. Besides that, you could just use a trowel or a shovel even, but that could lead to holes in your field or grass and could be a danger, so just be careful with that. So besides that, if you have any more questions or want to know anything else, please feel free to contact us at the information we'll be providing, and I hope you've learned a bit, and have a good day.